Bacteria are single-celled organisms. The plant pathogenic bacteria often have flagella as we see them here. So those are flagella. They allow the bacteria to move, especially in moisture. So in areas where we have more water on leaves and for a longer part of the day, the bigger risk we have to both bacterial and fungal foliar diseases. In this case, we have rhizobia on clover root, so these are beneficial bacteria that will allow the plant to fix nitrogen, to pull nitrogen out of the air and fix it through a chemical reaction that is occurring inside the bacteria with the help of the plant. Bacteria are single-celled microorganisms, and we cannot see them without a microscope. So we have both beneficial and detrimental. Detrimental means something that's not good for you. Plant surfaces and inside the plants. So if a bacteria is on the surface of the plant, we call it an epiphyte. If it's inside the plant, we call it an endophyte. Individual bacterial cells can only be seen with a microscope, and that would be under oil immersion. They are very small. We can see large populations of bacteria if they're growing on the surface of an agar plate, and sometimes we can see them on food that's been left too long in the refrigerator. So they differ from a fungal colony because the fungal colony is going to appear dry and thread-like or hairy, some people might say, where the bacterial colony is usually going to be moist and shiny. All right, fungi. Let's move on to fungi. Fungi are eukaryotes. Bacteria were prokaryotes, meaning there's no nucleus in the cell. Fungi are eukaryotes, and they digest food externally. We digest food inside our bodies. Fungi digest outside. And the broken down nutrients are absorbed through the cell walls and cell plasma membrane. Most fungi reproduce by spores and have a body that's composed of mycelium or hyphae. They are heterotrophs, meaning they get their food from something else, from another organism. Plants are autotrophs. They make their food themselves using sunlight energy. All right. So if a fungus gets its nutrition from a living host, that's called a biotroph. If it gets it from stuff that's decaying, they can call that saprophytes. Saprophytes or saprotrophs. If a fungus is killing the host cell to get the food, that's called a necrotroph. All right, here we have some examples of fungal diseases. The early blight of tomatoes we saw in part 1 was also a fungal disease. So there we've got black spot of rose, which is very common, Calototricum of English ivy, right? Phylosticta, and shot hole symptoms, right, on eggplant. So sometimes in a lesion we'll get what's called a chlorotic halo. So see, the lesion is the dark part or the black or the brown part, and around it is this yellow halo. So that's called chlorotic halo surrounding the fungal lesion. So chlorotic means a yellow, right? A yellowing of the cells, right? This is sorghum, right, which is a monocot leaf, and then this is citrus greasy spot. Oh, this is really good. This is cedar apple rust, and this is a gall that we're looking at on the cedar tree. So this is the cedar tree. This is the gall on a day that's not rainy. This is the gall on a day that is rainy. And so these little teleal horns have turned into a gelatinous mess and come out and are dropping spores. So this gall can live on this cedar tree for quite some time. And the spores released from these gelatinous teleal horns can infect an apple tree. Right? So if you have an apple orchard, you might not want these cedar trees around it. All right, so this is what the fungus looks like growing on a petri dish. So the petri dish is filled with agar, which is a sort of a jello-like substance, and we can transfer bits of tissue from a diseased plant or from a previous culture onto these agar plates and watch them grow. Here we have some color in these fungi. This one's kind of yellow. If we turn this petri dish over, we would see some red, and that's fusarium. So all of these are fusarium. There are many fungal diseases of roots caused by fusarium. All right, and then we have the powdery mildews. So these are very fascinating. This is a Cleistothecium, and it holds sacs of spores inside. So there inside is the sac, and it has 8 spores in it. This whole thing is a Cleistothecium. These are appendages. These fungi can be identified by the shapes that we find on the appendages. So these are fascinating. All right, let's do another slide. Okay, oh boy. So this is from a fungus called Monolinea, which causes brown rot of peach. This is a very significant disease of peaches. This is an overwintering structure. So last season, this peach fruit was infected with the fungus. It turned into what's called a peach mummy on the tree and dropped off the tree at the end of the year. So these cup-like structures formed over the winter. These are over the early spring, and these are lined with sacs of spores. So the spores are released from these apothecia. These are called apothecia, and that will infect the new peach trees. It will go into the peach flowers as they're blooming and infect the immature fruit. You won't see the symptom until the fruit ripens. It's a very, very common disease. It's very difficult to control, but we can clean these up. So this is a place where sanitation can help somewhat. If we clean up the orchard floor at the end of the season, we can get rid of these things and have fewer spores being released.